Okay, so thank you. Welcome to this presentation of Biomikas that is going to be hosted by Ifra Ferheen. Some information about Ifra is that she did a master in microbiology from COMSATS University in Islamabad in Pakistan. Sorry if I mispronounced that. And her master's research from the University of Camerino, focusing on biotechnological advantages from psicrophilic bacteria isolated from glaciers of Pakistan. Right now, she's in her second year of PhD as a candidate at University of Camerino, and one part of her research is performed at the National Institute of Health in Rome, Italy. So she'll be right now explaining a little bit more about her current research and what she's doing here in Camerino. So I will stop sharing my screen so you can share yours, Ifra, and we shall begin.
You can see my screen, Anna?
Yes, we can see your screen.
Okay, thank you so much for this lovely introduction. Hi, everybody. I'm Ifra Ferheen from University of Camerino, and today I am here to give my talk on a project which was performed in collaboration between University of Camerino and National Institute of Health, Rome, and the title is Plastic as Durable Islands for Harboring Multidrug-Resistant Bacteria in Aquatic Ecosystem. So first of all, I would like to give you a brief introduction of my topic. As we all know that plastic has become the commodity in our environment, and now we cannot imagine our life in a pre-plastic era. So if we just talk about facts and figure, annually 245 million tons of plastic we are producing, which is like 35 kilogram per person. And the dark part of this story is only 50% of this plastic is one-time use, like just use and throw type of plastic. So this use and throw behavior of consumer has elevated many problems, including like when we throw our waste after one-time use, somehow it escapes many recycling procedures and other waste streams and make their way to water bodies and marine environment. So the question is, what is the environmental fate of the plastic which is entering our water bodies and contaminating them? As we all know, the durability of plastic make it highly resistant to degradation, and under high environmental pressure, for example, tsunami, storms, or other or thermal pressure, sometimes this plastic material is degraded into small particles of 5 millimeter in size, known as microplastic. This microplastic is classification of microplastic. This microplastic pieces are categorized into two types. One is primary microplastic and other is secondary microplastic. Primary microplastic are tiny particles designed commercially in this size for some purposes. For example, if we talk about microbeads, they add microbeads into scrubs and other cosmetic materials, fiber of our cloth which we are wearing, and some pellets which are used in different industries. And on the other hand, if we talk about secondary microplastic are the broken pieces of the main larger fragments of plastic. For example, films, they are broken from the shopping bags which we throw normally, sponge which we are really using in our daily life in our kitchens. So whenever we are washing dishes, some pieces of this sponge is detaching and now they are entering into water stream and ultimately to oceans, fragments, and some other secondary forms. Let's talk about some facts and figures. Although plastic has so many benefits, we are using it in our daily life, but the only problem of this use of plastic is their when we are throwing it in environment, and it is creating a big problem. For example, if we talk about packaging, 40% of whole this plastic industry is consisting of packaging material. It's making plastic bags and other such packaging material, and the only working shelf life of these plastic bags and shopping bags are 15 minutes, not more than that. We just use it and then we throw it. 10 million tons of plastic are dumped in our oceans annually, and equal to more than a garbage truck per minute. And the worst part of whole this story is only 9% of this plastic got recycled. We have seen many environmental activists, many other NGOs, they are talking about reduce, reuse, and recycle. But still, a lot of public awareness is required on this topic, because only 9%, that is like nothing, which is recycled out of all this. All around the world, this topic has withdrawn considerable attention of researchers, and they have reported many research articles on how this plastic is affecting marine life. For example, they engulf it. Sometimes they are entangled by some plastic pieces. 1 million marine animals are like dying by plastic pollution. There is a foundation, Alan MacArthur Foundation, and they have claimed that there will be more plastic in our oceans than fish by 2050. And we are already in 2020, end of 2022. It means we have very short time to aware our public and to control this problem. What is, like, if there is a microplastic pieces in our aquatic ecosystem, what is the threat to aquatic ecosystem? So we know that fishes and the other marine animals, they cannot distinguish between microplastic and their prey. So often what happens, these tiny particles are swallowed by these fishes, and ultimately these we are consuming seafood regularly, and ultimately these microplastic are making their way to our plates. So what we are throwing in environment, after a complete circle, it's coming back to our bodies. It is in one research paper, it is reported that 40 pounds of plastic, which is huge, we are consuming in our average lifetime. So this study was conducted to answer our two queries. Number one, can plastic be claimed as specific vectors of pathogen dispersal, and are there any plastic specific pathogens? Number two, do plastic act as trafficking agents for antibiotic resistant genes? Selection of sampling sites. So our first goal was to decide where we want to perform this study. So for this purpose, we selected Bracciano Lake. This Bracciano Lake is located 32 kilometers northwest of Rome, of Roman part, and it's a caldera type volcanic lake. It is surrounded by three towns, Anguilla, Bracciano, and Trevigiano. And it's an important drinking water reservoir for the people of Rome and the residents of its surrounding towns. We use a geographical information system, GIS software, to select sites, because from which sites we want to collect sample. So for that reason, this GIS software provides us a significant amount of information of the land use around this Bracciano Lake. So we selected our sites on the basis of that. So total 7 sites across the Bracciano Lake were selected. Secondly, when designing our sampling strategy, we had 3 questions. Which type of bacteria we are focusing? Because we know in environment we have cultivable bacteria, non-cultivable bacteria, and a huge diversity. So we came up with a panel of targeted microbial community, which are human and veterinary pathogens. For example, Staphylococcus, Yersinia, Salmonella, Klebsiella. And for their isolation, we used chromogenic and selective medium. For example, here in the picture on the left corner, you can see on the rapid staph agar, black colonies of Staphylococcus. So at least morphologically, in the first step, we can say either we were able to recover some targeted bacteria or no. Second question was, which is the most suitable which is the most suitable weather for our of this type of study? Like, what is the impact of weather on microbial community of lake? As we know, in summer season, in spring season, when the weather of Rome is very good, people visit beaches very oftenly, and they do some water sports, swimming, and other things. So what is the effect of anthropogenic activities on microbial community of Bracciano Lake and the load of microplastics? So we selected we did three sampling campaigns in late summer, in winter, and in spring season. The third question, as we know, like different plastic topologies, different type of plastic polymers may have different effect, and they provide different surface for adhering bacteria. So for this reason, we shortlisted 4 plastic polymers that we are commonly using in our daily lives. So polypropylene, polyethylene terephthalate, polystyrene, and styrene acrylonitrile resins. So isolation of pathogenic bacteria from artificial plastic substrate. So this was our first strategy. As I told you in the last slide, we took 4 different polymers of plastic. We tied them with stones. We wrapped them in a net, and this whole thing was submerged in the euphotic zone of the 7 selected sites of Bracciano Lake. So we left them there for 1 month time period, which is enough for colonization of plastisphere. After 1 month, from all 7 sites, these plastic material were recovered and under sterile conditions was transported to a laboratory. Now the hard part came, the removal of biofilm from these plastic material. As we know, like, it was there for 1 month time period. So in that time period, many invertebrates, organic part, algae, and other things were also attached to this plastic. So we used scrapers and swabs to clean it and to recover as much as possible material containing colonized bacteria. Collected material was then pre-enriched in Mueller-Hinton broth, and this broth this was incubated at the temperature similar with the Bracciano Lake, that was around about 19 degree to 20 degree centigrade. And isolation and purification of bacteria was performed on chromogenic and selective medium. Single and isolated colonies were proceeded with biochemical and molecular identification. On the other hand, as I told you, like, we were performing a comparative investigation of microbial community in raw water and microbial community colonizing on plastic substrate. So from all 7 sites, simultaneously, we also collected 2 liters of raw water. We use 0.45 micrometer nitrocellulose filter. And these filters were then, we put these filters on chromogenic and selective medium. And similar procedure what we did with plastic isolates, we did on isolated and single colonies biochemical and molecular identification. We also wanted to check the load of microplastic in Bracciano Lake. So for that reason, we use another tool in the second half of the study that is known as Amenta, which is consisting of a net. On the other end of the net, there is a bottle. So it is tied with the boat, and boat take a round of the whole lake, and the collected material was filtered by using serial filtration method. So in this method, we use filter size or different pore size of filters, nitrocellulose filter, namely like 25 micrometer, 20 micrometer, then 5 and 0.45 to get rid of all organic matter, so we can clearly see small fibers, transparent sheets, and small fragments of plastic. Filters were dried at 65 degrees centigrade to remove all kind of moisture and for the better observation under the stereoscope. So we were also interested to check the antimicrobial susceptibility of all isolated strains from raw water and artificial plastic substrate. For this reason, we selected a panel of antibiotics that are commonly used in animal and human therapeutics. And so for all the raw water isolates and plastic isolates we obtained from different chromogenic and selective medium, they were proceeded with Kirby-Bauer disk diffusion assay, and results were interpreted on the basis of Clinical and Laboratory Standard Institute CLSI standards. So results. First, I would like to share some information about our site selection and how this geographical information system help us in selecting sites. So after using GIS, we knew that site 1, 2, and 6 were closely located to urban fiber, while the other 4 sites, BR04, 7, 8, and 9, they were more closely located with broadleaf forest, non-irrigatable lands. And I must mention here, like these 4 sites were had some beaches and some farmhouses, some cows and other animals were feeding there. So the microbial we were expecting a different microbial community from these 4 sites and the other 3 sites. So our results, our hypothesis was confirmed, and you can see in this graph, graph A is for the targeted microbial community from raw water, and the graph B is targeted microbial community from plastic. Here you can see BR0789 in both cases show more microbial diversity as compared to the other site, because more anthropogenic activities, human activities were carried out in these sites, as well as animals also impact microbial community. Secondly, one more starkly difference was observed in raw water microbial community and plastic community. If you see, there are some bacteria which were only recovered from plastic and not from raw water. For example, Citrobacter, Morganella morgani, Klebsiella, Salmonella, and some strains of Staphylococcus that were not recovered from raw water but from plastic. So after this point, we got an answer to our first question. Yes, there are plastic play an important role in amplification and dispersal of some pathogenic bacteria. And yes, there are some plastic specific pathogens. Secondly, we performed our samplings in sampling in 3 different seasons. That is end of summer, winter season, and spring season and spring season. And in the 3 samplings, we confirmed these results, like more starkly different microbial community was obtained from artificial plastic substrate as compared to raw water. Secondly, as I told you, we used 4 different topologies of artificial substrate and different plastic polymers. Here in this graph, you can see on x-axis different type of plastic used: polypropylene, polyethylene terephthalate, polystyrene, and styrene acrylonitrile resin. And on the y-axis, you can see targeted microbial community. So from this graph, we concluded like polystyrene and SAN were able to adhere more diversified bacteria as compared to PP and PET. If we just talk about Staphylococcus species, if you see here in the picture 2, from 2 sites, BR04 and BR06, we obtained similar results. So polypropylene and PET were not able to adhere more species of Staphylococcus, as we only managed to recover few colonies. But from PS and SAN, we obtained a large number of colony and a huge diversity. For example, Staphylococcus intermedius, captis, xylosis were recovered from PS and SAN, but not from PET and PP. Then antibiotic susceptibility test phenotypic profiling of the resistant isolates. So here in this graph, you can see 1 gram positive bacteria and 1 gram negative bacteria, and different panel of antibiotics for them. On the x-axis, you can see a panel of antibiotic tested. On the y-axis, you can see resistant isolates. Red color is depicting raw water and the purple is for plastic. So you will see in this graph, we can see different high resistant isolates from plastic as compared to raw water. Like, for example, if we take example of Klebsiella, Morganella, and Staphylococcus, 100% of isolates for these from plastic were resistant to all tested antibiotic as compared to raw water. Isolation of MPs and raw water of Bracciano Lake results. So dried filters were observed under the 1x magnification of stereoscope, and we observed red, blue, green color fibers, some film, and some plastic pieces. But this protocol was not optimized. Maybe we lose a lot of plastic pieces on Amenta's net, so we need to optimize this protocol. Conclusion and future perspective. Sampling strategy and methods for targeted microbial community characterization from plastisphere was optimized. Our results depict plastics can be claimed as specific trafficking agents for amplification and dispersal of pathogens. In nutshell, plastisphere communities starkly differ from microbial communities obtained from raw water. This study is not only for the public awareness, but also to draw the attention of authorities to aware public, like to apply specific measures to control plastic pollution in water bodies. Our future perspective of this project is microbiome analysis and detection of antibiotic resistant genes, that is resistosome, to investigate the potential role of microplastic in trafficking of antibiotic resistant genes, which is so our second question is yet to be answered. Thank you.
Thank you, Ifra. It was really interesting to hear what you've been doing and what you are still doing. I have a couple of questions, but I will let them to the end. So if participants want to ask something to Ifra, they can raise their hands, the little button, and we can let you speak. Open your mic, or if you want, you can also write your questions in the chat. If you're uncomfortable writing in English, you can write in Spanish and we can translate it, so Ifra also can answer you. Just a little minutes. Okay, Paul, do you have a question?
Okay, yes, I do have some questions, but first, thank you, Ifra. It was a fantastic talk. I know you were nervous because of the technical issues.
Oh, yes. And then when I shifted my presentation here, graphs were like not so good and without background. So that was the problem.
Yes, you changed operating system in 5 minutes and you had to learn on the go. But the presentation was fantastic.
Thank you so much.
Before I go with the questions, in the meantime, I hope some people start asking. I just wanted to, how do you say, make a point here, which is your work is dealing with two very, very important health threats right now worldwide. Yeah. So
yes.
One is relatively new. So microplastics. This is something relatively new that it's started to spread, and there are many works dealing with that, and that's important. And then you have another one which is older but is also important. So, and now your work is showing that maybe one is carrier of the other one. So microplastics could be delivering resistance to places where there should not be, right?
Exactly.
So yes, for all the audience, keep in mind this. These are two very, very important health threats right now for not only humans but also animals.
Animals, yeah.
And the work of Ifra is dealing with the connection between these two. So
yeah,
fantastic. Very, very nice.
I have a question about how did you could collect all this plastic from this lake that I forgot the name, but the lake that was near
Bracciano Lake.
Bracciano.
Bracciano, yeah.
So you have like a technique or a proper protocol you have to follow? Did you try to do a certain way so you can pick up more plastic with the microbiome or the microorganisms still around? How do you take care of your sample?
You are talking about which thing? Microplastic load or artificial plastic we put artificially in lake?
The ASP, the artificial.
Okay. So there is no, there is no standard protocol for that. We are like, I guess we are the first one who are trying to optimize it. So in literature, you will not find any study related to artificial plastic substrate artificially placed in any lake or any other water body. So this was the idea, like to simulate what is happening in the environment, because if you collect anything from environment, if I just pick up some plastic from Bracciano Lake, next time it's not reproducible. But what we did, we had everything under control, type of plastic, their time period of being in that lake or in that water body. So at least we are sure that what we are analyzing in our lab is reproducible. So for the reproducibility was literally a point in optimizing this protocol. But in literature or in any other platform, I don't think so you will find any standard protocol for this procedure.
Okay, nice.
Yeah.
Thank you. Wendy Tabucci, that is a student here in UPC, ask, what would you say they are the most difficult obstacles when dealing with plastic studies, research, or policies?
So the most important thing, if we are sampling microplastic, if you want to collect it from water body, we know it's like huge water body, especially if you are doing your work in ocean, even if you are doing in lake. So analyzing or estimating the real load of microplastic is really a tough thing, because what you are doing, maybe next time you are not taking round from there or some other factors. Microplastic collection from a lake, I guess, was a hard part. Second thing, everything is just colonizing on our plastic. For example, if this is polypropylene, everything is attaching to it, some invertebrate bodies and everything. So when we brought this material after 1 month to lab to literally extract everything, all type of bacteria which are adhering to it, was a critical point. So we had to clean it so many times, all parts. So we make sure, like, even if something is attaching here and not here, we should recover it. So to get good results, significant results. So these two things were so important and a critical point in our research.
Do you have like I don't know how to say it, like a cutoff point, like certain microplastics that are maybe too small, they just couldn't not be seen somehow? Or I don't know.
There is a possibility, because sometimes fiber is of white color or some transparent sheets. For example, the polythene bag sheets, in the small size. So on filter, white fiber you cannot see because it's already white background, you cannot see. So one thing that. Secondly, sometimes some invertebrates are transparent, their shell, their feather is transparent. So when you see under a stereoscope, you say, oh, okay, it's a microplastic. But when you touch it with the loupe, it breaks. But microplastic is not something easy to break. So it's a tricky point to distinguish microplastic from organic material. That's the tricky part.
You have to have a good eye for that.
Good eye for that and good experience. Like if you touch it with hot loupe, it should melt, not break. So there are some tricks to distinguish between organic part and between microplastic, especially the transparent one and something in brown in color, which is just like wood or straw. So yeah, that's the hard part.
Thank you, Ifra. I don't know if there are any more questions.
Me.
Yeah, sure.
Sure.
what idea are you thinking about on how these resistance genes got into the lake?
That's a nice question. So, number one thing, like what in our hypothesis, we cannot say 100%, but if I talk about few sites, many cows are farmhouses, like farm cows, which are like controlled, monitored for and for all kind of diseases. They got vaccines, they got antibiotics on regular intervals. So they are feeding on that site. So sometimes what happened, they are going there to drink water, they are excreting their waste material there. So their gut microbiota, it already contained that antibiotic resistant genes. When it is entering the environment, it is colonizing on the plastic, they are doing a communication and they are doing antibiotic resistant genes. Another project is ongoing to see if really these microplastic pieces are helping bacteria to uptake plasmid for environment, how they are communicating for antimicrobial resistant genes exchange.
Yeah,
So
this
I
is
think
and this is something that you could trace actually, right? So if you, for instance, sequence these genes, and then
yes,
sequence also the microbiota of the same genes at the level of the farms, and then you sequence the same ones at the level of the small river that gets to the lake, and this kind of things, maybe you could get a pathway, a real pathway of delivery, let's say, no? Maybe, I don't know,
exactly.
just an idea.
Yeah, that's true. We can do it.
Fantastic. Yeah, that was one question. And the other question was, why do these bacteria like to keep these resistance genes? One is, how do they get there? But why are they keeping them? I mean, they have to replicate more DNA. This is more energy and this kind of things, you know?
It's a tough question. So I try to answer it, but I think I'm not sure. I'm not 100% sure, but I try to answer it. So I think to cope out with environmental pressure, for example, if I just talk about Bracciano Lake, many streams are coming from different hospitals and other waste material in which extra antibiotics are releasing. So there is some concentration of antibiotic which is present in environment for sure. So maybe to cope out with such environmental stresses, they are keeping these resistant genes. For example, they also keep heavy metal resistant gene, even they are in normal environment. Maybe they are coming from some sewage and some other such kind of environment. So they try to keep it to cope out with environmental pressure, I think.
Yeah, this could be. Another alternative is super valid, your alternative. Another could be also, I mean, biofilm forming bacteria. They
Yeah,
love to acquire resistance and keep it for
resistance.
some
Yeah.
reason we don't know. And I think some of your data shows that those ones that are attaching to the plastic tend to have more resistance than the water
Yes.
counterpart, right?
So our like right now we are focusing on antibiotic resistant genes in plastic isolates and raw water isolates. So I didn't show that data yet, but yes, for sure, and isolates obtained from plastic contain many antibiotic resistant genes as compared to raw water isolates. So they are keeping it. Those are forming biofilm. They are colonizing on plastic. They are keeping antibiotic resistant genes. Yeah.
Yeah, and then I had another question, another tough question.
Mm-hmm.
Did
I
you
like
find
to answer questions.
any correlation between some bacteria that like to be with another bacteria in the same plastic sample?
Sorry, can you repeat it?
So if there is some sort of indication of something that some bacteria species
Mm-hmm.
like to be in the same plastic material as another bacterial species. So some sort of co-colonization.
I really don't know. No.
No,
I cannot
that
answer
would be
that.
cool, right? Maybe you have some sort of how to say, pressure or some sort of differentiation on some species. I'm thinking of something like the microbiota, you know, microbiome. You have many species and they are kind of working together. Maybe, since this is something new, microplastics in the environment, new for evolution, for nature, maybe they already found some way to stick together, right? And
Yes, but I think it's a hard part to say like they are always doing co-colonization or they are always together, because in environment we cannot depict. For example, from in all samplings we got E. coli, we got aromonas. So I can say like they are doing co-colonization, but they are in environment. So anything can adhere to that plastic. And like, for example, if I talk about Staphylococcus, we only got it one time, not the other two times, but rest of the bacteria in all three samplings. So that's why it's a little hard to answer. Yeah, but for sure there
yeah,
is.
only with single cell genomics, but this is too hard and too expensive. Very, very hard to do. Fantastic. I will stop asking you because I think there are more questions in the chat.
Okay.
Yeah, but very nice. Thank you very much, Ifra.
Thank you so much. Thanks a lot.
Thank you, Vol. Wendy Tabuchi is asking one more question. That is, what would you say it's important or necessary to expand this kind of research to other fields and work with multidisciplinary teams, including architects? I study architecture and I would like to know how can we get more involved with these topics. Same as Carla that started communication too.
Can you just repeat the first part, please?
Yes, would you say it's important or necessary to expand this kind of research to other fields and work with multidisciplinary teams, including architects and communication?
I think so. Like, it's not something really specific with biology or microbiologist or something, because first of all, being a human being, we have to save our planet. So that's the main thing. So everybody, even he's not educated, still he has to play a part to save our planet. We are already facing so many things just because of our own faults. Secondly, if we want to say different different fields, for example, architecture, they can, I don't know what they do exactly, but they can do a little thing which I am just thinking. Do not build buildings near to something that is nature. Like, for example, do not make a colony near to a lake. So you can preserve a lake in that way. So I think if that is the answer to that question.
Yeah, even make it safe to
make it like stop cutting trees, stop making buildings near to water bodies. Like, even I have seen so many apartments in Dubai and some other places, sea facing apartment, and they are advertising it. But if you are living just right next to sea, you are ultimately decomposing, you are throwing your garbage near to that, which is making its way to ocean. So I think if we want to stop it, we want to do some steps, architectures can do that. They make a agreement. We will not build anything near to ocean or other water bodies.
Thank you. Luciana Amado, that is part of our laboratory in Peru, asks, since these bacteria are isolated from microplastic, can we assume that some of them are also good candidates for plastic degradation? Do you have any plans regarding this?
Yes, it's true. For example, we managed to isolate Axiobacterium indicum from our plastic samples. And ultimately, during our literature research, we found out this is a plastic eating bacteria. It really can feed on it can use plastic as a carbon source. So yes, there are some plastic eating bacteria who were part of this microbial community. which was colonizing plastic. But in our current goals of this research, we are not focused on that, but maybe in future we will focus. Yeah, but there are for sure in literature, many bacteria, especially Axiobacterium, is reported as plastic eating bacteria. Yeah.
Have you seen this type of bacteria in your samples, or?
Yes, we were able to isolate Axiobacterium indicum in two of our samplings, I guess, from many sites. So we have Axiobacterium indicum, and we were just doing some literature research for all pathogens, which we isolated. And interestingly, we found out that this bacteria is not literally a pathogen, but a plastic eating bacteria. It can decompose plastic, but obviously to do that in lab, we have to optimize so many protocols. It's a long process. So right now we are focused on antibiotic resistant genes, the main problem, and not on degrading plastic. Yeah.
Thank you, Ifra. As Luciana says, cool. That sounds really, really cool. Okay, I don't know if there are any more questions in the chat, or you can raise your hand too.
That's it.
I think so, yes.
Mm-hmm.
Then I would like to thank you once again, Ifra, for your presentation, for telling us what you've been doing.
Really my pleasure.
Yeah, it's amazing, and I think what you are still doing, it's really cool. So thank you once again. Thank you everybody for joining. And okay, the students that are here also for the cultural passport points, we already registered you, so don't worry about that. And we will see you in the next Biomicas presentation next Friday.
Thank you so much for inviting me. Thanks a lot.
Thank you, Ifra. Thank you, everybody.
Thank you very much, Ifra. Very nice talk.
Thanks to you.
And super good luck in the last year of your PhD.
Thank you so much. This I need a lot. Thanks a lot.
You're doing great. Don't worry. Fantastic. Thank you very much to everybody. Thank you, Anita.
Thank you. Thank you, everybody. Buona serata.
Buona serata anche te. Ciao.
Ciao.